Let me begin by saying that Omicron was only reported exactly 3 weeks ago. And my team at the Aaron Diamond Age Research Center here at Columbia, as well as Harris Wong's team in systems biology and KYN's team at University of Hong Kong, along with our clinical colleagues, Michael Ying and Magda Sobiechik, responded immediately to see if we could contribute to the understanding of this emergent variant. And a lot of credit goes to everyone listed on this slide. And in particular, I like to acknowledge Li Hong Liu, Shou Ikatani, Yi Chen Guo, Lian Liu, and Jasper Chen for a tremendous effort, including many sleepless nights to generate the data in very short period of time. So first, let me speak about SARS-CoV-2 variants. This is a phylogenetic tree of many of the variants that you are undoubtedly aware of. Of course, the virus that started it all is the WU1 isolate from Wuhan. And of course, we see significant viral evolution, creating many different variants. And the Omicron variant, also known as B.1.1.529, is shown here in the red color. Last year, this time, we were just dealing with the emergence of alpha and beta variants. Alpha from the UK, beta from South Africa, and it turned out that alpha was more transmissible and beta was more resistant to antibodies. And in fact, as we know, alpha became dominant at the start of this year and reached our shores by late spring. And beta spread locally in South Africa for some time and compromised the effectiveness of vaccines that were made from the original spike protein. Some of you might recall that together with Ann-Cathrine Ullman's laboratory, we identified the so-called New York variant, also known as B.1.526 or IOTA. And that variant came and went more or less like Homo erectus. And it was basically dominated by alpha, which in turn was dominated by delta. And alpha was about 50% more transmissible than the original strain, and delta was about 100% more transmissible than the original strain. And hence, for the past 6 months or so, delta has dominated globally. And then the day before Thanksgiving, we heard about Omicron, initially detected in Botswana and quickly followed by detection in South Africa. And in South Africa in particular, it was noted to be rising rapidly. And within a matter of days, we knew that it had spread to many, many other countries. And over the past 3 weeks, we know it spread to over 60 countries. And in places like the UK, it's already becoming dominant with the doubling of cases every 2 to 3 days. So the projection is that this variant will take over in several weeks. And I would just say parenthetically that, again, work done by Hiroshi Mori in our group together with Ann-Cathrine's group, we're already seeing lots of Omicron at CUMC. The first batch only about a week ago had about 5%, and then next batch at 10%, next batch at 24%, and the most recent batch at 39%. So this is rising in our community, even when Delta is prevalent. The other thing, when Omicron was reported, that was striking was the extensive mutations found in the spike protein. And some of the mutations are quite familiar to us at position 484, for example, at position 501, 417. These were found in previous variants, particularly in beta, and were responsible for antibody evasion. However, as you could see here, there were over 30 spike mutations, including 15 in the important receptor binding domain. And this struck fear in all of us that antibody invasion by Omicron would be substantially greater than the beta variant, as listed here. And I think our study that I will now share with you amplify that particular concern. So when we began this effort, Harris's lab, particularly Li Yuan, was responsible for assembling the spike gene and to create all these mutations as quickly as possible, which they did with a few hiccups, but we got there. And then they made some of the individual mutations as well, with the results of which I will describe. And this is the dominant form of the Omicron. And you'll note that there is one additional mutation here that's seen in about 10% of the current Omicron isolates. And that mutation was added as well and characterized. So as this spike gene was assembled, Li Hong, Shou, and Maple in our group put the spike gene into pseudoviruses so that we could assess serum neutralizing activity against the original strain shown in blue and the Omicron strain in red. And initially, 10 samples from convalescent patients were examined. And these 10 came from individuals infected in spring of 2020, presumably by the original strain. And as you could see here, there were healthy titers against the original strain, and then one noted a substantial drop. In fact, 8 of the cases could not neutralize Omicron. And this basically represented a minimum of 30-fold decline in neutralizing activity against the new variant. And this is compatible with emerging evidence in South Africa that reinfection rate is at least 3 times higher with the Omicron than with previous variants. Next, we turn our attention to examine sera from vaccinated individuals. And you could see listed here are the 4 most commonly used vaccines: the Pfizer mRNA vaccine, the Moderna mRNA vaccine, the J&J adeno-based vaccine, and the AstraZeneca vaccine, also based on a different nano. And you could see that we already knew that the mRNA vaccines typically induce more robust neutralizing titers, and J&J and AstraZeneca lag behind. But you could see in all these graphs, every single case, there's a diminution of neutralizing activity when comparing Omicron to the original strain. This is over 20-some-fold at a minimum. This is over 8-9-fold at a minimum. And then, of course, these are, you couldn't calculate fold change. In fact, these 2 cases here for J&J were individuals who were not only vaccinated, but also infected. So one sees very, very dramatic decline. At Columbia, most of us got the Pfizer vaccine. So this is the profile. And you could see many individuals who were fully vaccinated. This is actually at the peak of the vaccine response a few weeks after the second shot. And you can see most have very low titers against Omicron. And of course, many of us now have gotten the boost. And so we assessed 15 cases who were not only fully vaccinated, but received a third shot of mRNA vaccine, either Moderna or Pfizer. Only 2 of the cases were Moderna boosted. The others were all Pfizer. And you could see that, once again, every single case, there's a decline in neutralizing activity against the new variant. And the fold change here is about 6.5. So these results, we believe, are compatible with data that are just appearing this week, showing vaccine efficacy is down to 30–40% with mRNA vaccines for fully vaccinated individuals, and down to about 70% for boosted individuals. These were just being reported from the National Health Service in the UK, as well as a private insurer in South Africa. The folks at University of Hong Kong in KYUN's laboratory, particularly Jasper and Hin, have been in parallel assessing the neutralizing activity of the vaccinees against wild type and B11529 using the authentic virus that they had isolated from a traveler that went from South Africa to Hong Kong. And as you'll find, as you'll see here, there in the live virus assay, there is also a substantial decline in neutralization against the Omicron variant. And in fact, most cases have no neutralizing activity against this variant. So the live virus data confirm what we observed using pseudoviruses. So next, we turn our attention to monoclonal antibodies directed to different classes of the receptor binding domain. Yi and Guo put together these figures. You could see that there's class 1. Class 2 is basically the receptor binding motif. And the mutations are shown in cyan. And the footprints of the antibodies are outlined, probably too small for you to see clearly. And these are for the receptor binding domain, and these two are for the so-called N-terminal domain, or NTD. And these are almost all the neutralizing antibodies are directed to these surfaces. And once you look at these pictures, you immediately see the risk posed to all these monoclonal antibodies, just based on the footprints in relation to the mutations. And indeed, when Li Hong, Shou, and Maple tested these antibodies against the wild-type pseudovirus or the B11529 pseudovirus for class 1, 2, 3, 4 RBD antibodies, you see class 1 antibodies essentially are wiped out. Class 2 antibodies are wiped out. Class 3 antibodies, one is modestly impaired, one is preserved, the others are wiped out. And class 4 antibodies, this is a region that is very well conserved among sarbecoviruses. Even those antibodies suffered a great deal of impairment. And the N-terminal domain antibodies are also knocked out. And as you may know, there are antibody drugs that have been authorized or approved for clinical use, largely for prevention, but one pair for prophylaxis. And those antibodies are also knocked out or substantially impaired, tenfold or more. One antibody that is used by itself is S309, and that one is impaired. But I mentioned earlier, there's a second Omicron variant that has a 346K mutation added. And when that pseudovirus was built and tested by the same folks here, you see pretty much the same picture for class 1 and class 2 monoclonal antibodies. And now for the class 3, all of them are down substantially. And class 4, similarly, and the NTD antibodies. So this is essentially a wipeout of all the available monoclonal antibodies that exist today. Obviously, there are many more that we haven't tested, but in general, these antibodies represent what's been isolated and characterized in the field. Lastly, we turn our attention to focus on the specific mutations that affect, confer antibody resistance to Omicron. The results that I just showed you could be summarized in this heat map on the top 2 rows. And basically the 346K variant of Omicron is wiping them all out. And when we assess the individual mutations, you notice several things. One is at position 493 here, which is affecting class 1 and class 2 antibodies, class 1 antibody, class 2 antibodies, by knocking out hydrogen bond and causing steric hindrance here. And then 2 additional mutations at position 440 and 446, causing steric hindrance and impairing antibodies in class 3. And then finally, there's this interesting mutation at position 371, changing a serine to leucine, which in certain conformation may, in the down conformation, may interfere with the glycan at this position and altering the overall conformation for class 3 antibodies. And in the up position, it may affect this loop, which is recognized by many of the class 4 antibodies. But how this mutation could also affect class 1 antibody and it is really not known. So this is the most interesting mutation, which would need to be further characterized. Lastly, Shou and Li Hong just did the same panel antibody on not just the Omicron and the second Omicron variant, but also on Alpha, Iota, the California variant, the Delta, Gamma, and Beta variant, just to demonstrate how Omicron resistant relate to the resistance of other variants. And you could see the Alpha variant was not very resistant to antibody, even though it spread very well. Then came these additional variants that basically resisted the NTD antibodies as well as class 1 and class 2 antibodies. But now with the Omicron, you have even greater resistance to class 1 and 2 antibodies, but the emergence of resistance to class 3 and 4 antibodies. So it's not too far-fetched to think that the SARS-CoV-2 is only a mutation or 2 away from pan-resistance to currently elicited antibodies. So the conclusion, our conclusion at this point are the following. Omicron is extremely resistant to antibody neutralization. It is threatening vaccine efficacy and flattening the antibody therapy landscape. The loss in serum neutralization is likely the result of resistance to all classes of antibody to the spike. We identify the 4 mutations that I noted. And basically, for the past year, we've been chasing after the virus, one variant after another. I think we need to jump ahead and anticipate the evolutionary trajectory of this virus and develop newer strategies, particularly ones that could target conserved elements. So I will end there, and thank you again for the opportunity to share these results. And I have to once again acknowledge a group of dedicated people who have worked so hard over the last 3 weeks.
Thank you, David. The very sobering remarks. We'll open it up for questions. So let me start with one. So it's not clear how all these mutations accumulated. Is this because the virus was in some way hidden in a population that was poorly sampled, and therefore acquired mutations that were largely neutral until some mutation emerged that significantly increased its infective rate? So the companion question is, do we now have enough data from infected people to assess whether pathogenicity has also remained, the same or increased, or in fact, some have reported has actually decreased?
Well, 2 comments. One is that if you look at the branch on the phylogenetic tree, it's very long, and then it's as if it may have developed in one or a few individuals, or one or few animals. We don't know. There are lots of speculation out there. I guess a lot of people would put their money on the fact that it may, since it was first detected in Southern Africa, where HIV prevalence is great, it may have persisted in one individual for a long time under the selective pressure of different antibody responses. And this we have seen in our immunodeficient patients. It's been reported maybe a dozen times. But of course, that's speculation. There are also a few signature mutations found in this virus that are shared with viruses that are passaged through rodents. And so there are some folks who think it may have gone into animals and come back to human. There's really no evidence to concretely support any of these hypotheses. In terms of pathogenicity, it's really hard to tell right now. The initial outbreak in South Africa affected largely young people, and so the pathogenicity was low, and a lot of them were also reinfections. So it's hard to judge. And now, given the high prevalence of vaccination, we're seeing infection in vaccinated individual, which you would expect to be less pathogenic. So we have to wait and see, I think, in terms of its lethality.
Thank you, David. I see a question from Malamaini.
Hi, thank you for a really clear talk. I wanted to know a bit more about your thoughts about future strategies as a more resistant immunogenicity approach, and particularly whether you think the S2 region, which recent work suggesting might elicit more pan-coronavirus protection, might be an interesting region.
Yes, there are antibodies directed to the so-called fusion peptide of S2, and they could neutralize, but the neutralization potency of those antibodies are so much lower. So it's hard to see how they would protect unless you find a super monoclonal, which we have yet to see. Obviously, this directs our attention to protease and polymerase and other targets that are known to be better conserved than the spike. And of course, we now already have small molecule drugs to the protease and polymerase. And I think a lot more work along that line should proceed. And in terms of vaccines, I think we have to jump ahead and see if we could, it is a very, very broad antibody responses. And there are many, many groups putting their thoughts together along that line.
We have a question from a unidentified participant, 44188552. If you can please identify yourself.
Lily Yamasaki.
Thank you. Ask your question.
Yes, hi. Wonderful talk. Could you comment on, compared to HIV, how much of a problem the glycan shield could be in SARS-CoV-2 versus HIV in terms of antibody development and finding neutralizing epitopes?
My overall impression is the glycan shield is much more prominent than HIV.
Wow.
And the glycan, there are a number of glycans on the spike protein, but we find antibodies binding to virtually every surface that's available on the RBD, as if it's unobstructed. And there are glycans that are part of the epitope, particularly so-called VH1-2 class of antibodies. But my overall impression, I wouldn't be an expert on that matter, but my impression is HIV glycan shield is much more prominent.
The HIV or the SARS-CoV-2? Sorry.
HIV.
HIV, okay. All right. Thank you very much.
Question from Ryan Kasner.
Hi, David. It's Ryan from Larry's lab.
Yeah, hi.
Do you think that the Pfizer and Moderna will make a new Omicron booster? How hard would it be to redo those clinical trials?
They're already making it. They started almost on day one of Omicron detection. In terms of how well they would work to elicit an Omicron-specific response, we have to wait and see. I think many groups are trying to do experiments now to see if we're going to face the problem of the so-called original antigenic sin. I think a lot of us have seen the original spike, and whether a boost with a heterologous spike would elicit the right antibodies, that is not known yet. That was tried with the beta variant without that much success by Moderna.
And John Hahn?
So about a week ago, the Times had a piece saying that South African researchers had found a couple of patients who had not well-controlled HIV, who had, over the course of months, this many mutations, same relative approximate number of mutations, that they tracked developing in their bodies during that time period. Do you view this as that kind of situation as a likely source of Omicron?
That I would say is the leading hypothesis, but I don't think we could ever prove that at this point. I think, we have here at Columbia, we have seen, for example, the virus replicate in a transplant patient on immunosuppression for 4 or 5 months with the virus continuing to evolve in ways like this. And so it's quite conceivable, but whether this one emerged that way or not is not known.
So David, just to close it, because then we have to move on. So is a new clinical trial required when you're just adding one variant to an existing vaccine, or you think there will be a shortened, safety profile analysis?
I think that, it depends on what the preclinical data show. If preclinical data show that boosting with a new vaccine, Omicron-based vaccine, would elicit the right antibodies to neutralize this new variant, then I think FDA has already said that mRNA platform technology would allow the companies to move very quickly.
So you would treat it as a phase 4, essentially.
Essentially, I think they're suggesting that it could be substituted within a few months.